Hello everyone. Previously I have done the PYQ series from Unit 6, that is plant hormones, sensory photobiology and secondary metabolites. Now those three subunits are something that from where questions are always expected. You cannot avoid those units. Now when I say 6C and 6H, that is nitrogen metabolism and stress physiology, questions, it's not that questions are not expected. Questions are expected from these topics also, but not on a regular basis. It's like a rotational basis. Sometimes they ask, sometimes they ignore. So overall, if you take into consideration the entire Unit 6, the topics which is not being asked on a regular basis are nitrogen metabolism and stress physiology. But I have taken a few questions just to make you understand how the questions come and how you need to approach the questions from these topics. So the very first question is, the following statements are made regarding the nitrogenase enzyme involved in the reduction of atmospheric nitrogen to ammonia. Nitrogenase enzyme is composed of two components, dinitrogenase reductase. Molybdenum iron protein component is a homodimer. Iron protein component is dinitrogenase. Molybdenum iron protein contains the active site metal cluster where the nitrogen binds. Iron protein delivers the electrons to molybdenum iron protein component in a reaction coupled to the hydrolysis of magnesium ATP. That is hydrolysis of ATP in presence of magnesium ions as a cofactor. So you need to identify the correct combination of statements out of this. So here the correct answer is statement A is correct, statement B is correct and statement E is correct. So why the statement B and C are incorrect? Let's have a look onto that. Nitrogenase enzyme complex is separated into two components, that is the iron protein and the molybdenum iron protein. Remember, neither of which has the catalytic activity by itself. That means individually they are not active, but when combined together they can be activated. So the iron protein component of the nitrogenase is smaller than its other component and is an iron sulfur protein which is extremely sensitive to oxygen and irreversibly inactivated by it. So this iron sulfur protein is a dimer of two similar peptide chains having a molecular mass of 30 to 72 kilodalton. So this is a homodimer because it is made up of two similar peptide chains. Now molybdenum iron protein has 4 subunits and each subunit has 2 molybdenum iron sulfur clusters. So they are different. And this component is also sensitive to oxygen. So that's the reason statement B and C were incorrect. Next, usually the process takes place via redox reaction. So we have got the reduced ferredoxin. So this is the small component iron protein, large component molybdenum iron protein. So remember, it is in small subunit the ATP hydrolysis takes place. That is 16 molecules of ATP are used. So this ferredoxin, reduced form of ferredoxin reduces the iron protein, which in turn reduces the molybdenum iron complex. That helps in the reduction of nitrogen, giving rise to ammonia. Moving on to the next type of question. Now this kind of question is a match the following. That means they are provided with you a list of microorganisms and their symbionts. You all are aware that biological nitrogen fixation can be carried out in two ways. Either it could be free living organisms or it could be symbiotic organism. So you need to identify the correct match over here. So to get into a correct answer, you have to be very clear with this table. So as you can see, this is the category of symbiotic nitrogen fixation. So leguminous plants, the host plant has many symbionts. It could be azorhizobium, bradyrhizobium, mesorhizobium, cynorhizobium. So remember, everything ends with rhizobium. Actinorhizal family, they have Frankia as their symbiont. Gunnera to nostoc. Azolla to anabina. Sugarcane to acetobacter. Miscanthus to azospiralium. So these are some examples of symbiotic nitrogen fixation. Now compare it with a free-living nitrogen fixation. We have cyanobacteria. Example, anabina, calothrix and nostoc. Some bacteria can be categorized as aerobic, facultative and anaerobic. Aerobic means that are depending upon oxygen. That is azospiralium, acetobacter, bigernica, duxia. Facultative, that can survive in both the condition, whether oxygen present or not present, it is fine. Bacillus and Klebsiella. Anaerobic microorganism includes non-photosynthetic and photosynthetic bacteria. So non-photosynthetic, Clostridium. Photosynthetic, we have Chromatium. Now here you can filter out few organisms that are commonly used as, they can be used as either a symbiont nitrogen fixers as well as a free-living nitrogen fixers. Now that categories includes nostoc, anabina and azospiralium. And also not to be confused with the terms acetobacter, azotobacter. So azoto comes under aerobic free-living bacteria. Aceto is a symbiont for sugarcane. So these are the few things to be considered. So based upon this match the following, you can easily get into the answer. Okay. Next question from the same topic is, following are certain statements regarding nitrogen uptake and assimilation by plants. So plant roots can take up nitrogen in the form of nitrate or ammonium ions. Ammonium ions taken up by the plants can be assimilated into amino acids. Amino acids are exclusively synthesized in plastids and chloroplast of roots and leaves respectively. Nitrate can be stored in the vacuoles of both roots and leaves. So what is the correct combination of statements here? So you have statement A is correct, B is correct and D is correct. So why C is incorrect? Because amino acids are not exclusively synthesized in plastid. So if you look into this image, so this is the summary of the nitrogen assimilation that happens. So you have got nitrate getting reduced to nitrite. Nitrite enters inside the chloroplast. With the help of some GS-GOGAT mechanism, ammonia is synthesized. Then ammonia will help in the production of amino acids. So here it is synthesized in the chloroplast. Now this glutamate in presence of aspartate aminotransferase can be synthesizing aspartate. So that takes place in the cytoplasm. Aspartate can give rise to asparagine. Asparagine can give rise to other amino acids. So it is not mandatory that the amino acids are synthesized exclusively in chloroplast or plastid. It can be other locations as well. For example, cytoplasm. Moving on to the next question. Now this is from stress physiology. So here they have provided with some abiotic factors with their corresponding effects. You have to identify the correct combination. Water deficiency. Now this is related to water potential reduction. So whenever you get these kind of match the following questions, try to get a confidence on a particular combination. So when you get one combination perfectly, if you are lucky enough, that will be the only combination found in the entire 4 options. So that will help you out. Okay. Sometimes, for example, many of you are aware high light intensity is related to photoinhibition. So that you are confident. So you are getting two such options here. You can easily eliminate these two options and try working out these two options. So this is also one way of tackling match the following kind of questions, because match the following kind of questions are mark fetchers. If you get, you will be getting a 4 marks as a total. So all you need to do is have a patience to read the question thoroughly, read the combinations thoroughly, and whatever you have studied, try to recollect. Salinity, now this is related to iron cytotoxicity. Flooding, it is in that condition where there will be hypoxia situation comes to the roots, right? So that becomes the correct combination here. So water deficit stress can be defined as a situation in which plant water potential and turgor potential will be reduced enough which can interfere with the normal functions. Salinity will have the similar aspects to water stress excepting for the addition of iron cytotoxicity, that is, salt is present excess in soil. When the soil becomes waterlogged or flooded, the air spaces in the soil are filled with water, restricting the diffusion of oxygen to the roots. As a result, what happens? The roots will be deprived of oxygen. Obviously, so that condition is known as root hypoxia, or we can also say anoxia. Then photoinhibition, it refers to the reduced photosynthetic activity and damage to the photosynthetic machinery due to excessive light exposure. Of course, light is required to carry out photosynthesis, but if there is a high light intensity, it can result in damage of the photosynthetic machinery. So that will affect the process as well. Next, so this is also one kind of question, match the following. You can also expect statement based questions as well. Here, some statements are provided with relation to osmotic stress in plants. Accumulation of ions during osmotic adjustment is predominantly restricted to the vacuoles. In order to maintain the water potential equilibrium within the cell, other solutes such as compatible solutes or compatible osmolytes accumulate within the cytoplasm. Galactose is considered to be one of the compatible osmolytes involved in osmotic stress in plants. And there are mainly 4 groups of molecules which are commonly used as a compatible solutes. So what is the correct combination over here? Statement 1, 2 and 4. So that means galactose is not an example of an osmolyte. So remember one thing. Galactose is not working as an compatible solute or an osmoprotectant. Rather, as I told you, you have 4 major osmoprotectants present. That includes mannitol, sorbitol, glycine betaine and proline. So just imagine, if you have studied only, if you know about only the osmoprotectants, you can easily fetch a 4 marks out of this. So overall speaking, when I say unit 6, it is obviously an important unit from examination point of view, with a strong focus on 6A, B, D, E, F and G. C and H, I am not saying you cannot get questions from here, but the frequency is less and not always you expect. But remaining all the 6 subunits, you can expect questions every time. One or two, you can expect questions. So overall speaking, you get good number of questions from this unit. So prepare accordingly and do practice more questions from this topic. So that will help you in boosting confidence in yourself. So that's all from my side for today's session. See you again in another session. Thank you so much.